Awesome. All right. I'm going to close down the participant in the chat list, but if anyone does have a question and I am not calling on you online, hey, feel free to just like speak up because I won't necessarily be able to always see it. All right. Oh yeah, you're go for it. You can turn off whatever you need. Just don't fall asleep on me. Yeah. All right. My biochemistry warriors. Welcome. Do you want to do the opposite maybe? Yeah, the front and then turn on the back. Is that okay? Will we pass out? Okay. All right, good. All right. We are here to embark on our 7-hour journey of biochemistry. It's going to be fun. It's going to be great. We will break for an hour for lunch, partially for my brain and also partially for your brains, and so you can get some fuel. So at 12, we'll take a break for an hour, and then we'll come back. If we finish up early, we finish up early, although we are scheduled to be here until 5 today. So we'll just kind of see how the day takes us. You are not required to be here. This is something that is just a value add for you. If you find it valuable, great. If you don't, I'm sorry. But so stay as long as you like, leave whenever you want, get up, get down, eat snacks. Just try not to do anything super disruptive. So no like HIIT workouts in the middle of our day, please. I've seen it all here, you just never know. And really how I have things structured today. So last night, those of you who joined me, I appreciate those of you who are here in person and those of you who are here online. I know 6 to 8 is late, especially because we haven't started term yet. But I really wanted to make sure we got 4 weekends in before your actual test. So that's why we did start this weekend. So every Friday for the next 4 weeks from 6 to 8 p.m., I will hold an in-person practice exam. It's simulated to feel kind of like an NPLEX. So it's the full 2 hours. You have 2.5 hours technically to do 100 questions. So this shortened time really should push that timing of 1 minute per question, which is our goal for NPLEX, is 1 minute per question. These practice exams are focused on a content topic. So our first weekend, we're focusing on biochem, whole weekend. So that's all we're doing today. Next weekend will be anatomy focused. We'll do a little bit of embryology, a little bit of histology in that. The third weekend, we'll do micro and immunology. And then the last weekend, because this tends to be where people do the best on our exam, is physiology path. We'll just really lean into high yield for physiology path. So some of the areas that get a little wonky, or some of the concepts that are maybe can get confusing or that are commonly tested on that we should review kind of right before our test, our exam. So Fridays from 6 to 8, we'll always hold an in-person practice exam. I'll always post them online. So if you're like, I don't want to stay, or if it's snowy or cold or icy or whatever, you can take the practice exam at home. And I encourage you to take some Scantrons with you. I'll always have them up here. If we run out, let me know and I'll bring some back, but they'll always be kind of housed in 107, or you can grab some in the faculty office, admin 250. But I encourage you to practice your Scantrons now. It's just like one less thing that you have to think of that's different on that day, right? One less thing you have to worry about. Then Saturday sessions will be content-based. This weekend for biochem, it's a lot of fill in the blanks, a lot of path systems, drawing things out, highlighting, circling. Anatomy will be a lot of, we'll have some pictures that you'll label, but that's not how the test is. So I'm also going to kind of have us stretch and convert to cases. So we'll be trying to do that conversion for anatomy of, okay, what is this structure on a picture and how can we identify it? Keep that picture in our brain, and can we apply this now to just words? Because the anatomy portion on the test is tricky because we don't get images. And we learn anatomy here, it's very image-based. So that will be next weekend. And then our microimmuno will go through all the major bugs and their diseases and the things we really need to know. And then the path physio will kind of fall into place. We'll always have a lunch break from 12 to 1 on our Saturday sessions. There's Gigi. So 12 to 1 will always lunch break. I believe that every other weekend we're like 1 hour shorter. So next weekend for anatomy, we go till 4. And the last weekend, I think we go till 4. We'll just kind of see where we go. If we need to stay a little bit longer, hey. If we need to cut it shorter, that's fine. Okay, so Moodle page. All of you should have access to this Moodle page. This is the Moodle page for specifically the basic science board review for spring. If you don't have access to it, it's probably because you didn't get officially registered through Caligari in the registrar's office. So feel free to send me an email or a text message if you don't have access to this page, and I will get you added to this page today during lunch. Why? Because this page is where I'm posting all of our content for this weekend. Eventually, it'll make its way onto the general page that typically happens once I close down this course. So if those of you who have accessed the general NPLEX study page in the past, this will eventually get uploaded and added there, but it won't probably for the next month. So I would really encourage you to focus here, at least for these sessions. Has the dates and the times, and then I will post kind of the breakdown schedule so you know the content topics. That allows you to kind of pick and choose what you want to stay for. We kind of went through briefly this review introduction yesterday, and then we did our practice exam. The practice exam that we completed yesterday, just so you all know, those of you who want to take it, was the full NPLEX I biochem practice exam. It's 100 questions, so it was relatively long. Those of you who are here completing it can probably attest to the mental fatigue, the brain fatigue of 100 biochemistry questions. And then the answer key is posted as well with the same name, just answers under a different folder. There's 2 other practice exams. This one's a half, so it's only 50 questions. And then this one is another 100 question test. I'll post probably 2 or 3 more full biochem practice tests. I probably won't get them up until Monday or Tuesday because I have to record for structure and function today and tomorrow. But I will get them up for you. I have them. I just need to get your answer key done. So you'll have some more biochemistry practice. I told you yesterday, and I want to reiterate this, at this point in the game, 1 month out, you really should be high yield, moderate yield studying. So that means practice questions, flashcards, practice tests, discussing things with your friends, mind mapping, but really trying to do as much as you can without looking at notes right in front of you. So from your memory, and then checking yourself, checking your answers. That will get you the best high yield for these type of exams. Not as much reading or highlighting anymore. When we go through our lectures today, the first lecture we go through will be this review of biochem exam. I pulled 25 questions that I think are important that we highlight, and they're not necessarily covered throughout the rest of the day as well. The rest of the 75 we'll touch on throughout the day. So we cover these first 25 in this review, and then we'll get into kind of our part 1, part 2, part 3, part 4, part 5. You'll see that all of these parts have a handout, and the handouts for students, they are meant to be like worksheets. You'll see that they have blanks in them. That's on purpose. You aren't having a defective form. It's so that you actually can practice watching this lecture or going through these on your own and filling in the blanks, almost like a study sheet, like a workbook. You have access, though, to the exact PowerPoint that I'm presenting on today, too. It's located just right below. So if you want to go through the lecture today, the exact lecture that I'm giving, you can pull up the PowerPoint. If you want to go through and try to fill in the blanks as we do, go through or print things off during break, whatever works for your brain to study, great. If you're a doodler like me, you might want to fill in the blanks because it might help you to doodle along and write things out as we talk. But it's really just a way that you can kind of go through the content. All right, I think that's all my logistics stuff. Online, are you able to hear me okay so far?
Yes.
Awesome, thank you. I know you can't see me via video, correct?
Yeah, we can.
Okay, all right. I actually prefer that, so that's great. Awesome. I want to use a video star. Okay. Any other questions from those in the house? Okay, cool. Well, we'll get into this review then. If you haven't taken the exam yet, that's okay. We're only going through a few questions. I do have these subtitles on here because I do tend to talk fast and sometimes slur my words together. So just ignore them if it bothers you. If you're watching the video later, you can actually move the video down if you don't like seeing the subtitles, and then they're out of the screen. All right. So when you score the exam, I encourage you to go through the examination, score yourself. If you have already, great. If you haven't, that's totally fine. You can do it later. The exam score guide, all of the answers are online, so you have access to all of that. If you scored, this is kind of like how you can grade your success on this first practice test and really any of them moving forward. If you were below 50%, so less than half the answers right, that's a below average score, that's okay. We're just starting out this month, okay? But I encourage you then to review biochemistry content, supportive materials. I posted Khan Academy videos they're a little bit under the level of what you need to know for like an NPLEX test, but it's a great foundational place to get started. So if you're having trouble with some of these biochemistry concepts or some of these metabolic pathways, Khan does like break them down in a way that's easy. And then you can kind of take that next level of, oh, what are the cofactors? What are the regulatory enzymes? What's the rate limiting step? So you can kind of take that next level. You'll then want to transition study to your moderate and high yield from there. If you're getting between 50 to 60%, that's actually an average score on these tests. So that you're right where you should be. So if you got half or a little bit more than half right, start to transition study to moderate high yield concepts for the next month and take more practice exams, just more repetition. If you're between 60 to 70%, you're likely on track to pass within a month out. And if you're greater than 70%, continue what you're doing. You're likely on track as well. So with this in mind, again, any score is fine. We're just starting our practice here, but you kind of will know where you're at depending on where you scored on this practice test. You'll see the anatomy will have a similar breakdown. And then when we get to 2 weeks out, this will shift a bit. And we're really hoping that we're hitting between the 60%, 70%, or 70% and above when we're about 2 weeks out from the test. That's typically on average what we need to see to have a successful test rate for NPLEX. Okay. Doing good so far? Okay. I talk fast, so if I go too fast, like tell me to slow down, like you can do the pace, or if I'm going too slow, like tell me to speed up. I can go faster. All right, so I'm starting towards the beginning of the test and working my way forward. So this was question 96. The question is, what would be the interactions between the side chains of aspartate and arginine at neutral pH? And our options are hydrophobic, ionic, hydrogen bonding, steric, and covalent. It's a classic biochem question using amino acids. They love asking about amino acids because there's 20 things we can look at, right? So here's our aspartic acid and our arginine. So we know here aspartic acid is an acidic amino acid. Arginine is a basic amino acid. So with that in mind, what do we think their interaction is going to be? Hydrogen bonding is a good one. I like that option. What would be another option we could consider too? Ionic, great. So hydrogen bonding and ionic should be your top two, right? So hydrophobic, we're going to eliminate because they both have charges on them. So we're not necessarily thinking hydrophobic. We would really be thinking hydrophobic if we're talking about the hydrophobic category of amino acids, right? Steric, these aren't necessarily, this one is kind of sterically inhibiting. It has a really long side chain, so it could have some steric hindrance, but we most of the time think of that for our aromatic amino acids. And then covalent bonds, those are those like, permanent or more permanent, really strong bonds with an electron sharing. And we're not really assuming that we're doing any of that here because we don't have an enzyme. We're not pushing any reaction forward. So these are the two best answers. Your correct answer is ionic bonding because we're talking about the reaction between the side chains here. This acidic side chain is going to have a negative charge at neutral pH. This basic side chain is going to have a positive charge at neutral pH. So they are going to interact via ionic bonding. If this was not if we're not talking acid base here, if we were just talking in an alpha helix and we're looking at amino acids, what type of interaction would happen in an alpha helix? Hydrogen bonding would be your best answer. So these should be the two pieces that pop into your head. Great. Oh, I gave away the next one, but that's okay. In the formation of the alpha helix, the carbonyl group, so our carboxylic acid group, our C double bond to O, is connected by what? And we can see an image here. So we already know it's going to be connected to our amino group with that hydrogen, because I gave you the picture. You didn't have this picture on the test. So we could say, okay, it's this carbonyl group's connected to an amine group, that hydrogen bonding here, like you mentioned. So here's the example. So we can eliminate peptide, eliminate disulfide. So we're just left with A, B, or E. These are all of our hydrogen bonds. So we're saying, okay, is it a neighboring amino group, but it's a distance kind of far away? Is it a neighboring amino group we don't have any distance far away? Or is it just a single amino acid on an adjacent strand, just general? Given these three options, which one feels like something you could eliminate of these three? A, B, and E. Which one's different? A is different because it talks about 4 amino residues, sure. What's the other different one? Two of them talk about amino groups, right? A and B talk about amino groups. E doesn't. They all three talk about hydrogen bonding. So all three of these, I'm like, okay, all three are good answers. E doesn't talk about amino group, and I'm seeing here, I'm given now, I know it's an amino group, so I can eliminate E. So I'm dealing with A or B. So then we have to think about alpha helix. What's that structure, right? That circular helical structure. When we're dealing with stabilizing an alpha helix, the secondary protein structure, are we going to stabilize with things right next door to each other? Or would it be stronger if we're across from each other and we like grabbed hands? Would that be a stronger bond? Or be stronger if I held hands with the person next to me, if I'm trying to stabilize a circular structure? First one, across, right? That across bonding is going to be much stronger for that circular helical structure. Now, if we're talking beta-pleated sheet, different, right? But directly across is going to be more helpful. So if I'm making a circle, I'm not really going to be able to strongly hydrogen bond with my next door neighbor. That's not going to stabilize my alpha helix. I'm going to be looking at someone across from me. So then we get into the correct answer being A, because it lists specifically that 4 amino acid residues from it. If they said 5, that'd also be correct. If they said 3, that would also be correct. But the idea is that it's not the next door neighbor, it's someone across from me in the alpha helix. So that's what we're looking for here. So if given a question about secondary structure, alpha helix or beta pleated sheet, think about what that structure looks like. Then look at what the question's asking you. It's asking about a carbonyl group. So, okay, that's our carboxylic acid group. What type of bond, who would it interact with? Well, if it's an oxygen, slightly negative, it's going to be attracted to something slightly positive. So probably a hydrogen. So probably hydrogen bonding. So then there's your bond. And then who is it going to interact with? Next door neighbor, someone further away. The further away would be the best answer. Okay, that's how we approach these questions in NPLEX. You're going to get NPLEX, you're always going to get questions you've never seen before, right? Because that's the nature of the exam. So we have to kind of walk our way through logically how we could get there. And this is just showing 1 amino acid, 2 amino acids, 3 amino acids, 4 amino acids, and then that's that bond between 1 and 4. And I just tracked carboxylic acid groups. Okay, 88. We're now moving to enzyme kinetics. Okay, what statement about Vmax is true? So Vmax means what first? When I say Vmax, what am I referring to? Just think about what Vmax is, the word. Maximum of, what's V? Velocity, max velocity, great. So max velocity, and I said enzyme kinetics is what we're dealing with. So the maximum velocity of an enzyme kinetics. So what's an enzyme doing? What's its job? Catalyst, pushing forward what? A reaction, right? So we're talking about the velocity of this reaction, this enzyme, this generic enzyme is pushing forward. So which one is true? Is Vmax independent of enzyme concentration? We'll just read through them, then we can logic our way. So Vmax is independent of enzyme concentration, okay? For Michaelis-Menten enzymes, the maximum velocity is approached asymptomatically, okay? Sounds weird. Vmax can be attained only when half of the enzyme is bound to the substrate, okay? The reaction velocity is maximal when the concentration of the substrate equals Km. The reaction velocity is half Vmax when the entire enzyme is bound to the substrate. Okay, reading these questions without any guide can feel wonky, and NPLEX doesn't give you an image. So this is the image I want you to think of, is just this picture here. And you don't actually need to have the multiple lines. The line I want you to pay attention to is the black line here. The black line is your normal enzyme kinetics. So when talking about Vmax, we're going to see that Vmax or velocity is related to substrate concentration. That is your classic enzyme kinetics graph that you're going to see. So we can then rule out A, right? Because we see that velocity is directly plotted against substrate concentration. Or actually, whoops. So that was enzyme concentration. There we go. That was enzyme concentration versus substrate concentration. So velocity is directly plotted versus substrate concentration here. When we look at our enzyme kinetics, they mention the reaction velocity is half Vmax when the entire enzyme is bound to the substrate. So if all of the enzyme present is bound to substrate, right? If we have full enzyme substrate binding here, we're fully bound. We've used up all the substrate, all the enzyme, all the substrate present that we can. Are we looking at half Vmax? No, we're looking at full Vmax because we've used up a lot of our substrate concentration. So we rule out this one. Reaction velocity is maximal. So we're saying we reach Vmax when the concentration of the substrate equals Km. Well, Km, its actual definition, is half Vmax. So we know that's not true. And we'll look at what this looks like in an equation here in our first kind of presentation today. We'll talk about enzyme kinetics. So if this is all not sounding familiar, don't worry. Vmax, velocity maximum, can be attained only when half of the enzyme is bound to substrate. That's saying exactly what D said, just a different way. So that's saying exactly kind of the same thing. So we cross that out. For Michaelis-Menten enzymes, the maximal velocity is approached asymptomatically. Kind of sounds weird, but I don't know if that's right or wrong necessarily. I can't prove it's wrong, so I'm going to leave it for now. And then Vmax is independent of enzyme concentration. Is that true or false? Why? Absolutely, more enzyme would speed up the reaction. If we have more enzyme, it can bind more substrate and it's going to move things along quicker. So it is not independent of enzyme concentration. So the only one left is B, and that is the correct answer. So when I looked at B and read B, I was like, that sounds weird. I don't like that. I'm going to eliminate it. Sometimes the weird one's right. You have to look at what the question's asking you. Can you rule out the other ones as being false? We could. So then we're left with B, which is true. Okay. And this is a great example of an NPLEX question. All right. What is not a second messenger? Another great rule out question. So our second messengers, we have kind of our two classes, our G protein coupled, and then the ones that are dealing with our inositol triphosphate group. Okay, so we have kind of these two separate categories of second messenger systems. So in our G protein second messenger system, our goal is to often get to cyclic AMP. So we know that this one is a second messenger, so we can cross it out. And we can assume, if you didn't know, if cyclic AMP is a second messenger, cyclic GMP is also one. So we can remove these two. And that is true. Both of them can be used as second messenger systems. From there, we have to remember our phosphate, our triphosphate, our inositol triphosphate second messenger system. And that's the one up here. IP3, inositol triphosphate, and DAG, diacylglycerol, are the two second messenger systems in this system. So E and C are also second messengers, leaving us with D being the not second messenger. Where some people get kind of freaked out is D is this, PIP, phosphatidyl inositol biphosphate. So PIP2, that is D. This is a receptor. It's a protein in the membrane. It's a transmembrane protein. So it's actually what is going to contact with the G protein during this reaction. It itself isn't sending a message, though. It is contacting this G protein, and from there, it's going to, then we get the activation of our second messenger systems. But this itself is static in the membrane. Does that make sense, the differentiation between the two? It'd be like adenyl cyclase is not our second messenger in our second messenger system here. Cyclic AMP is. Adenyl cyclase is a transmembrane protein. It's going to interact with our G protein that's going to translocate from the initial receptor over here, but it itself is not a second messenger in the second messenger system. So that's where they try to trip you up. If you had no idea what this is, great. You probably chose it, no problem. Now you know what it is, so you can be more confused. No, hopefully that makes sense. All right, there we go, second messengers. And then PIP. All right. One more kind of general, and then we'll move into a few other pieces. In what principal transport forms is nitrogen transported from the muscle to the liver? Well, to go through this one, we have to think about, okay, well, so when we're transporting something from muscle to liver, what's probably broken down? Protein, a macromolecule, right? Because muscle, that's a lot of proteins. We probably have some protein breakdown, so some protein byproducts. So what are our protein byproducts going to be? Amino acids. Those are the building blocks of proteins. So all of these are amino acids. That doesn't really help us answer the question, but that gets us into the right set of mind. Because then we're thinking, okay, protein breakdown, amino acid degradation, or protein degradation, what are our byproducts going to be that then get pushed into the liver to really get us fuel, right? Because that's most of the time when we're breaking down protein, we're trying to get some type of fuel most of the time. We're trying to reuse those amino acids for something else. So we'll go through the process of protein breakdown later today, but this is just a little mini one here for you. So we have in the muscle, we're using glucose, we get pyruvate, and then we are deciding, oh, we actually need to get more glucose and pyruvate into our liver. We're in a fasting state or an exercise state. So the muscle during protein breakdown takes that pyruvate and it converts it to alanine using alanine aminotransferase. So alanine is going to be one of the amino acids that's transported from our muscle to our liver. So if you knew alanine was one, that's the classic, you could eliminate C, D, and E. You'd have just A and B left over. And then you're dealing with, is it glutamine or leucine? The other one, and we can kind of see this here in muscle protein amino acids, ammonium, glutamate. We can kind of say, okay, well, glutamate's involved. Like, that makes me feel like glutamine should be involved, but let's make sure you are in fact correct. So when we take in our proteins, our amino acids, and we have these extra amino groups, one of our amino harvesters is glutamate or glutamine. And so glutamine is the other protein breakdown amino acid that gets then transferred, shunted over to the liver. So alanine and glutamine are your correct answers. This is probably a memorization one, unless if you're gonna know and really feel confident about protein degradation, protein breakdown. When we look at this, it'll kind of sink it in. But alanine and glutamine are going to be your nitrogen transporters from muscle to liver. You could also think about alanine and glutamine are going to be your best nitrogen stabilizers from your muscle to your liver. When we talk about transaminases, ALT, AST, right? They both have A's because they both contain alanine, right? So we can start to kind of make these clinical physiologic connections here too. All right, if we take that next step. All right, so those were the basics for the exam, kind of going through. This will be our first PowerPoint that we go through this next hour. We kind of covering those concepts, macromolecules, bonding, amino acids. Now moving into our review of the exam, these are some that involve more of the pathways of biochemistry. Your glycolysis, your Krebs cycle, your pentose phosphate pathway, your oxidative phosphorylation, all those good fun things. So question 62 from our practice test. What amino acid residue catalyzes the transfer of a phosphate group in the course of one of the steps of the citric acid cycle? Did I know this? No, I had to find this myself. I had no idea. Okay, so full transparency here. But if we are looking here, I gave you the actual reaction that this takes place in, and it's one of the reactions we don't typically care about in the TCA or the citric acid cycle. It's citrate to isocitrate using a conitase. It's the movement, it's essentially movement of this group here. So we're going to see some interaction of this group here to here. And the actual amino acid that stabilizes is histidine. And so we can see this interaction here. I would have not known this had I not looked this up. So this is something that this would be like one of those NPLEX questions of you've never seen it before, right? Why is histidine a good stabilizer? Well, histidine has some interesting pieces that we'll talk about, but histidine is one of the best amino acids to be a buffer in physiologic pH because it's pKa, its ability to kind of titrate its acid-base content is around 6 point something. So it's really good buffer, right? It also is relatively small molecule, even though it has this kind of funky side chain. And this funky side chain allows it to hold an extra positive charge. So it's pretty small. It's easy to fit into small places. So it tends to be a good stabilizer molecule, a good stabilizer amino acid if needed, because it can bind really easily and then break away. Its binding is not super strong. So it can be kind of bound for a moment. Its affinity is relatively weak, and then it can move off if not needed anymore. So we tend to see it here. Cysteine with that sulfur bond, that disulfide bond, there's nothing in the citric acid cycle that really leads me down the road of wanting to talk about disulfide bonds at all. So that leads me away from cysteine if I was trying to like logic my way through this. I might have been pulled with serine because of the hydroxyl group, right? That seems like a good stabilizer, so that could have pulled me away. Although it doesn't necessarily have a positive charge to stabilize, but it didn't really talk about that. Just talk about phosphate group. And then glutamine and glutamate, again, more acidic. I don't know, OH groups. I don't know how I probably would have logicked through this outside of just knowing histidine's small and typically a good buffer. That's probably where I would have gone. All right. What is one way NADH transfers across the mitochondrial membrane? This is number 55. So what is one way NADH transfers across the mitochondrial membrane? So I gave you an example here, but we have some listed. So does it transfer hydrogen from mitochondrial NADH to cytoplasmic NAD+? Does it introduce NADH by the GAP or glycerate 3-phosphate shuttle? Does it introduce electrons from NADH by the glycerol 3-phosphate shuttle? Does it transfer electrons directly from NADH to FADH2? And does it introduce H+ from NADH via DHAP, dihydroacetone phosphate? Dihydroacetone phosphate and GAP or G3P here are both from glycolysis. Those are both byproducts of glycolysis, the breakdown of glucose. So that's where we get these pieces. When we're talking about NADH, there's two shuttles it can take. One shuttle does a 1-to-1 transfer of electrons. One shuttle actually reduces electrons across the mitochondrial membrane. It's why we have a little bit of a subtraction of ATP when we go through our metabolism process. So if we look here, the example I'm showing you is NADH. We have our dihydroxyacetone phosphate, our DHAP, moving to glycerol 3-phosphate, G3P. This is a reaction in glycolysis. And then we are coupling that reaction with a creation of FADH on the inside of the mitochondrial matrix. So this is, in fact, the shuttle that's used. It's going to be the glycerol 3-phosphate shuttle. So that gives us two options here. Are we going to introduce it directly, or are we going to introduce the electrons indirectly? And if you look at this shuttle that I'm giving you on this picture, what would you say? Is it going to be directly giving NADH inside the membrane, or are we just transferring the electrons inside the membrane? Electrons, why? Anyone can popcorn up. There's no wrong answers here. I mean, there are wrong answers, but there's no wrong answers that are bad. Every wrong answer is learning. They get reduced, okay? So we're seeing here that we take our NADH, we're moving it to NAD+ on this side. Is there any NADH on this side, on the actual inner mitochondrial matrix? No, there's none. We're not actually shunting over NADH in this shuttle. What we are is we're energetically shunting over that NADH via electrons. So we're taking NADH and we're giving the electrons essentially to FAD. So we're getting FADH2 on the inside via this coupling. So that's the shuttle, that's the shunt here that we're seeing. We actually will come to find out NADH gives us about 2.5 ATP, FADH about 1.5 ATP. So the shuttle that's used, or the shunt that's used for NADH to FADH2 from our NADHs that are created during glycolysis, if it has to use this shuttle, which we'll talk about when it has to, we're actually minusing 1 ATP that we could generate from a full oxidative phosphorylation. So it does have an energy cost, but otherwise we wouldn't be able to get those electrons inside the mitochondria. So remember, glycolysis takes place in the cytosol. And all of our actual TCA cycle, our electron transport chain, oxidative phosphorylation takes place inside the mitochondria. Can you hear me again online? Yes. Okay. I'm sure you can. All right. Yes. Great. Awesome. My mic died. I didn't forget about you. Okay. All right. Now, moving from glycolysis to glycogen. What type of bond is glycogen phosphorylase able to cleave? So we're looking at, if we remember glycogen, we have two types of bonds, an alpha 1,6 bond and an alpha 1,4 bond. They are just referring to the carbon numbers that are bound together. Okay, so these carbon numbers are going to be 1 and 4. These carbon numbers are going to be 1 and 6. So 6 being this little doodad, 1, 1, and 4. That's the best I can write. So if knowing glycogen has alpha 1,4 and alpha 1,6 glycosidic bonds, anything saying beta, we could get rid of, right? Those are easy eliminations. Then we have to decide, is it alpha 1,6 or is it alpha 1,4? And that's when you need to actually know the enzyme in question. So we can get down to, in this instance, we can get rid of 40% of options. So get down to a 60%, was that 30% chance of getting it right? Yeah, 33% chance of getting it right by eliminating 2. And then this glycogen phosphorylase, I don't remember. Oh yeah, okay. I was like, I thought it was 1,4, but I was like, glycogen phosphorylase is going to break down alpha 1,4 glycosidic bonds. So if you knew that, you can eliminate these 2. All right. If you didn't know that, you're probably going to have to make your best educated guess from here. So this would be when knowing the enzyme will help you, but you can often at least get down a little bit lower in your answers before. So D is the correct answer. It's going to break these alpha 1,4 glycosidic bonds. It's going to break this bond here at the glycogen branch. So this is where the branch point is. So it's going to break the normal chain where it branches, but it's breaking the 1,4 bond. That's the breakdown. All right. Yes. There is the one that breaks the 1,6. We will cover it. I don't have it at the top of my head. Yeah, absolutely. Now, I do not have all, I should full disclosure, I don't have all these pathways memorized. I do have all of them down for you. Absolutely. But I don't have them all in my brain. There's just no way. Except for when I took the NPLEX, then I had them somewhere. No, they happened. All right. Next question will be question 44. Remind me to come back to it if I don't lean into it when we get there. Okay. Question 44, the first step of beta oxidation. So I have beta oxidation here. First step, once we have our fatty acetyl-CoA, our first step is going from here to here. We're dealing with an FAD or FADH2 that should scream to you oxidation reduction reaction, right? Anytime you see an FAD and NAD, I want you to think oxidation reduction, redox reaction, all right? If you see the hydrogens on there first, right? So if the hydrogens are on there first, let me just, okay, yeah, there you go. You're going to have a reduction of whatever thing you're interpreting or whatever you're dealing with. So if you're dealing with FADH2 to FAD, the actual thing that it's affecting is going to be reduced. There's going to be a reduction, okay? Because FADH to FAD is being oxidized. Does that make sense? If FADH to FAD is being oxidized, the thing that it's interacting with, the other reaction it's coupled with, is going to be reduced. Because we always have a redox reaction, reduction and oxidation together. And we'll look at that here in our next presentation, just generically redox. So we have an FAD to an FADH. So this now is not, this itself, is it getting oxidized or reduced? The FAD to FADH. Reduced. So whatever it's interacting with is going to be what? Oxidized. Great. So we know we're at an oxidation. This is obviously if you saw this pathway here, right? We're just working through this pathway that we get to see, whereas you won't get to see on the NPLEX, but we get special powers in our review session. And now we know it's an FAD because we see there's an FAD right here. So it's going to be A. Okay. When we learn beta oxidation, we're going to learn a pattern. Okay. It's going to be an oxidation, a hydration, an oxidation, and then a cleavage. Okay. So we're going to learn this pattern. So if you have a question like this, you can at least get down to a 50-50 split. Then you just have to know if it's FAD or NAD that's doing the oxidation first. So the best thing for some of these fatty acid breakdowns or buildups is learning the pattern, not necessarily being able to like step by step write it out like we might for glycolysis or Krebs. What prosthetic group do all aminotransferases contain? So aminotransferases, right, are aspartate or alanine aminotransferase. They're all going to contain one prosthetic group. The prosthetic group we have here is actually peroxidyl phosphate. So your answer is D. This is an example of when we need to know our vitamins. And so cofactors and coenzymes, they're just things I encourage you to memorize. The best thing to do here is going to be repetition, repetition, repetition. I have at the end a list of the key enzymes with the vitamins and their processes. So we will look at that together as a group at the end. But this is a straight memorization. You're going to need to group these together and know what cofactors and coenzymes, what vitamins are needed for certain enzymes, specifically rate-limiting enzymes that will be asked on NPLEX in some way, shape, or form. Peroxidyl phosphate is vitamin B6 or a component of vitamin B6. What metabolite is required for the synthesis of both purine and pyrimidine nucleotides? The answer here is going to be PRPP or phosphoribosylpyrophosphate. pyrophosphate. So no one wants to say that, so we just call it PRPP. PRPP is the rate-limiting step in that synthesis. Oftentimes, if NPLEX is going to ask you about a step in a pathway, it's going to be on the rate-limiting step and the rate-limiting enzyme. So we're going to lean into those when we talk about pathways. We'll identify the rate-limiting enzyme or the rate-limiting step, and we'll know the substrate and the product of that step and any cofactors, if relevant, involved. Okay. So PRP synthetase is the enzyme involved here, going from ribose 5-phosphate to PRPP. Okay. And this is the creation of purine and pyrimidine nucleotides, which are going to eventually lead to what? What are they going to build up into? Our purine or pyrimidine nucleotides. The backbone of DNA, the backbone of our genome. Yeah, absolutely. All right. The last few questions here, and then we'll break, are about more pathologic connections, pathologic biochemistry. So a little bit more clinical. Sometimes questions like these are counted under the path section, so disease and dysfunction. Sometimes questions like these are counted under the biochemistry section. So it's really hard to say. It depends on kind of the total percentage of what they use for NPLEX, but we still want to understand and make those connections when we can. What is a result of a genetic disorder in which the alpha chain of hemoglobin is not produced in sufficient quantities? So hemoglobin has two different types of chains, alpha and beta, and it has 2 copies of each, right? It's our classic hemoglobin molecule. So when we're dealing with, okay, hemoglobin chains, we can say, okay, I know there's alpha and beta chains. So this automatically draws my eye in. So I'm like, whoa, that has alpha and beta in it. So then I just need to know, okay, well, which chain is being affected? Well, it tells me the alpha chain. So my brain is like, I'm going to go pathologic resistance, alpha thalassemia, and that is the correct answer. So sometimes it really is what you think, or is the most obvious response. Don't psych yourself out. So alpha chain hemoglobin not produced in sufficient quantities, that is, or quantities, that is thalassemia, and we're dealing with alpha chain because it told us we were. Two different types. Correct sequence in protein digestion. I just gave you the answer here because this is one of those really long questions. You had to read all the different answers. They all had kind of different orders. These type of questions, when they have like these long processes and procedures, it's best to read the question first, kind of in your brain, walk yourself through what you think the logical answer is, then look to see if any of the answer choices match what you know is true, because they can try to throw you with answer choices that look right but are wrong. So when we deal with protein digestion, when we eat protein, it goes into our stomach first. So the first thing that's going to happen is we're going to hit the low pH of the stomach and pepsin in the stomach. Stomach's the first thing that encounters that protein. From there, that's when we can get our hormones involved. So from there, that's when we get cholecystokinin, a hormone that gets released or gets activated in the pancreas. Because hormones were released from the stomach that signaled to the pancreas, hey, protein's coming through soon. We have acid building up in the stomach because we're digesting this thing. So then pancreas releases digestive enzymes into our small intestinal lumen, right? That's where they go. From there, we have oligopeptides are formed by the pancreatic proteases because we have to first have the activation of the pancreas. From there, then we can actually have our oligopeptides formed by our pancreatic proteases. We break down the proteins further, right? By the protein breakdowners, the protein enzymes. And then we get to our brush border. On the surface of our intestinal cells, that's when we actually get down to individual amino acids, is at the brush border. So stomach churning acid starts to break things up. Pancreas is activated, secretes stuff into our small intestine. That breaks things into oligopeptides. Then we get to the brush border on the small intestine. That's where we get down to final amino acids, and then they can get taken into the cells. Okay. You know all of this. It's just when you see it all written out and there's 4 different options, you start like confusing yourself about which one's true. So that's why I walk through it first before I look at the answer choices, just logically. This is literally a question from a structure and function exam that I gave last term. I didn't realize it when I had this question on here, but so, yes. What is the source of energy for muscle contraction during seconds of intense sprints? So we're talking about a combo of muscle physiology plus biochemistry. So when we're sprinting, what type of energy we need? Do we need fast energy or slow energy? Fast. We need it right then and there. Do we care if we have oxygen for a long period of time when we're sprinting? Not necessarily, right? If we're doing a 100-meter dash and we're Usain Bolt, we're going to finish under 10 seconds, right? We don't need oxygen for that long. If you're hopefully one of my high school athletes this year, you're running it in like 12.5 seconds. I'm going to state. It's great. We'll see how they do. You're like me running like 14 seconds. But anyways, all right. So source of energy. It's going to be fast energy. So we're dealing with seconds here. So we really have two options. We have ATP standalone and creatine phosphate. Creatine phosphate is actually a molecule. I think I may have a picture, I may not. But it's a molecule in muscle. We have about 10 seconds worth of creatine phosphate in our muscle at any given time. Essentially think of it as a storage form of ATP. So it's not just ATP standalone. We only have like 1 or 2 seconds of ATP present in our muscle at any given time. It's not in a storage form. But we have a little bit longer, like 10 seconds of creatine phosphate, maybe like 10 to 15 seconds, enough to get us through a sprint. So because ATP would be used up too quickly, we'll cross that out. Our best answer is creatine phosphate. Now, if I was talking about endurance running or distance running or even moderate intensity sprinting, we would not probably want to use either of these methods for ATP, at least for the longer duration of our workout. We might use them up first. We'd probably be more relying on aerobic metabolism if we were able to get adequate oxygen, or anaerobic metabolism if we were becoming oxygen deprived. And that could happen in distance running or in really any type of exercise that's prolonged duration if we're not getting adequate oxygen supply in. So if we work out for a long enough period, or if we work out at a high enough intensity for a long enough period, okay? So thinking about the duration of exercise they're asking you, and then what would be the easiest source of energy that lasts for that duration. That's how you would approach this question. Your glycogen stores will be used up more in your minutes to hours of exercise. Typically they won't pull them in seconds. But if you last over a minute, then you'll start pulling from your glycogen stores. All right. Hemolytic anemia, 48, question 48, can be caused by a deficiency of which enzyme? This is just a you know. If you are in IV therapy shifts, has anyone been on an IV therapy shift? Are you allowed to yet? Some of you have been. Okay, what is the thing that we always want to check for? G6PD, yeah, absolutely good. I'm glad you got there because I didn't lead you down the road really well. G6PD, so when you go through environmental med, you will learn that you need to check for a glucose 6-phosphate dehydrogenase deficiency, right? You want to make sure that you don't have an issue with that enzyme. Why? Because if you do, you can actually lead to hemolytic anemia. And what can increase our likelihood of going down that pathway? Well, anything that can increase our reactive oxygen species. When we think about IV therapy, what's the thing that we often are worried about that can push us down that pathway? What's the thing that we infuse? The high dose vitamin C, yeah. So that's what we test for. We test if we're doing vitamin C, anything above, I think it's like 10 milligrams or 10 grams or so. I think it's 10 grams. 10 to 15 grams. Okay. I don't ever remember what it was. I just knew that better to be safe than sorry. If there's anything else over 5, I just tested for it. So I was like, I don't want to get sued. But yes, any high dose vitamin C, we worry about increased reactive oxygen species, which could be good, right? We're trying to do some antioxidant harvesting, so that's like a good thing. But if we don't test for this G6PD dehydrogenase deficiency, then we could potentially lead to hemolytic anemia, our blood cells actually lysing. And that is because G6PD is needed in this pathway to go from glucose 6-phosphate to 6-phosphoglucolactone. And really, why do we care about this pathway? It's the pentose phosphate pathway. It gives us NADPH. NADPH, think you need it for any time you're dealing with reactive oxygen species. And so antioxidants, I think NADPH. Hydrogen peroxide, I think NADPH. Respiratory burst, I think NADPH. We need good, adequate stores of this around because it's involved in all of these reactions in the body to help us resolve these kind of ROSs, these reactive oxygen species. Okay, cool. Some clinical stuff, good times. All right, deficiency of what vitamin is indicated by methylmalonic acid accumulation? So methylmalonic acid, you're like, where does this happen? It's in your homocysteine metabolism piece. We'll look at this towards the end today when we look at vitamins. But homocysteine, just think vitamin D or vitamin B12 and folate. Anytime you think homocysteine, those are the 2 vitamins I want you to couple in your brain. And the vitamin that's involved as the cofactor for this reaction is vitamin B12. Vitamin B12 can take methylmalonyl-CoA to succinyl-CoA, which then can go back into the Krebs. If we don't have adequate B12, it's going to be shunted over to methylmalonic acid. So it's going to be shunted over into this pathway of MMA accumulation. And that's something we can actually test for to see if you have a vitamin B12 deficiency or a vitamin or a folate deficiency. So we can measure some of these pieces to see, is this an issue with folate or is this an issue with B12 or is it an issue with both? So that's clinically why it's relevant. B12 is our vitamin that we are concerned about. Huntington's disease, good old genetic pathology. Because remember, genetics is in the biochem section. It's not quite super heavy, but genetic pathology can be there. So this isn't just talking about symptomology or saying genetic mutations, specifically asking you down to the weeds of like what's happening with Huntington's. It's a good one to memorize and know the mechanism. And I'm just going to tell you, it's expansion of trinucleotide repeats. This is like the classic example of this type of pathology. So if I was going to go through and start memorizing genetic SNPs or mutations or types of mutations, I would try to just do a 1-to-1. So this would be my example of trinucleotide repeats. Down syndrome is your example of trisomy, right? So I would try to make these individual connections with these genetic mutations. I wouldn't go much further down the weeds than that in regards to genetic pathology. But Huntington's is often asked about. So trinucleotide repeats, this is the healthy gene, CAG, CAG, CAG. This is an unhealthy gene. So we have this trinucleotide repeat that goes on for 37 to 80 repeats. So it's essentially, it's almost like they did the copy paste and they put their finger on it and just kept going. And so then we get this degenerative disease that is autosomal what? Nope, but that's a good guess. It always is, except for this one. Dominant, yeah, autosomal dominant. So this one, if someone has this condition, Huntington's disease, and they had offspring, that offspring would also have Huntington's disease. Why do we not see Huntington's in all the population then? Why isn't it everywhere? Why isn't it rampant? It's kind of a sad reason. Kind of, yeah, naturally. Oftentimes, people who have Huntington's disease do not actually last long enough to reproduce, or their symptoms are so severe they're unable to reproduce. So it is literally like a natural selection kind of component. They aren't unable to reproduce, or ethically, if they know they have Huntington's disease and they haven't developed symptoms yet, they don't want to have offspring because they know it will pass to their offspring. People that get it then, they've often came from a person who has Huntington's who hasn't developed symptoms yet, or who maybe had some symptoms but hadn't been diagnosed and didn't do genetic testing. That's oftentimes how you see that continue down the lineage. Yeah. I love those. It can. So the question for online was, as the number of trinucleotide repeats indicate severity of disease, they found in some cases, in some instances, it can indicate that, this like the severity of onset and the kind of progression through the disease, but it's not like a one-to-one. It's not completely true for all cases. So I wouldn't lean into that. Yeah. Yeah, it's a good question though. All right. And then last, and then we'll break after we get through, there's just a few more, are like true genetics. What's the function of helicase? Helicase, is it removing stuff? Is it eliminating supercoils? Is it synthesizing DNA? Is it adding negative supercoils, or is it separating the strands? Back to your molecular biology. What does helicase do? It opens it up, it breaks hydrogen bonds, and it unwinds the strand for us. So it's going to utilize the energy of ATP. Okay, thanks NPLEX for giving us extra words for separation of strands. That's what you're looking for, right? Unwinding of DNA. It has to use energy to do so. So fine, that's logic. But it's breaking hydrogen bonds to open up DNA. What's the nature of contact between DNA and the TATA box binding protein? It's my favorite thing, or TATA box binding protein. So the TATA box binding protein is not the TATA box, right? TATA box is what? It's in your DNA. It's kind of like a promoter. It's essentially telling all of the DNA machinery and machinery, this is where I want you to replicate the genome. So that's what it's like, your identifier. It's almost like a tag. So kind of like a promoter, like a tag. So the TATA box binding protein binds the TATA box, right? But the TATA box is part of DNA. So it's part of the genome. So it's actually, they're just saying here, the TATA box binding protein binding to the TATA box, which is DNA. So they don't really care about where or what's happening. They're asking, what's the bonding relationship between these two? And so what happens is when your TATA box binding protein binds to DNA at the TATA box, it's going to cause a kink in the DNA strand. And that's the target, that's the sign to DNA polymerase, hey, this is where you're going to bind and start replicating. That's how it knows to bind and replicate, is it actually changes the conformational status of the DNA strand. Okay, so if we know this, TATA box binding protein, you know it now if you didn't know it before, binds to DNA, and it's causing an actual conformational movement of the DNA strand. What could cause that? What could actually force something to move away or move towards something else? Like a full molecule. Could like a single bond do that? Probably not likely, right? A single bond, like an ionic bond or a covalent bond or even complementary binding, probably wouldn't cause like a full like form to move, a full protein molecule, a full molecule itself to like move shape. It's pretty aggressive. So then we're looking at like hydrophobic or protein-protein interactions, kind of generic. And your actual best answer is hydrophobic interaction. If they had steric hindrance, I would like that. That would feel really good too. It's not correct for this answer, but that would be harder for me to choose between because steric hindrance can kind of move molecules out of the way. But it's actually hydrophobic interactions. And you can look at the chemical composition and explain why, but it doesn't really matter. Hydrophobic interaction, if stuff is moving away from, right, we see that TATA box binding protein binds, and then the DNA is like moving away from it. It's like, oh my gosh, get me away from you. And it's because there's hydrophobic competition, right? Things pushing away from each other, not wanting to interact and bind together. That's your best answer here. So if I was answering this question, I probably would get to here, and then I would like maybe do a 50-50. I don't know. This one seems like it could have pulled me as a detractor just because I didn't know what that meant, right? And then question number 1. This one was kind of like, it was tricky, but it wasn't tricky. It's tricky because it almost felt like a trick question. But what if we're replicating DNA, how do we replicate DNA? Not think about this, but just normal replication. Normal transcription, sorry. What's normal transcription? What direction do we go? 5 to 3, right? Always 5 to 3, just like in your head. Okay? I'm telling you, reverse transcriptase, same way. 5' to 3'. Okay? Same way. even though it's going the opposite direction. We are reversing the transcription, right? We're going from reverse transcriptase. What does that mean? We're just going from, instead of going from DNA to mRNA, we're going from mRNA to DNA, right? Reverse transcriptase, reverse transcription. Typically done by what? By humans? No. HIV, viruses. Yeah. So, yeah, but specifically HIV is the one that we classically think of, right? And so we'll lean into this more when we talk about bugs in 3 weeks, but the answer is exactly what you would think it is, but you probably psyched yourself out because you wanted it to be different. Yeah, absolutely. That's exactly why I had this question as the first question, because it's such a great NPLEX question. It's exactly what they do. So 5' to 3' end is the direction of reverse transcription. Okay, they switched here. C is also tricky because it's 5' to 3', but they have hydroxyl to phosphate end. I want you to know 5' is always the phosphate end. 3' is always the hydroxyl end. Okay, 5' has a phosphate, 3' has a hydroxyl. Right, so they just tried to switch those words to see if you actually knew that the phosphate was first and the hydroxyl was second. Bummer. Okay. Whew. Popcorn to start us off. All right. Let's take a break. Let's take a 10. Get up and stretch, get some water, bio break, whatever you need. When we come back, we'll go in part 1. Okay. 10-minute break online, folks. I'll look at your questions now. Okay, great. Awesome. Any questions?